ये सबसे पहले वाला
बायस लॉगिन नहीं किया किसी ने, कर लो आप वो करते जाते हैं।
हाँ सर मैं कर हाँ हाँ Okay, so good afternoon to all of you. So I think few people have joined. So I'll just start the class. So this is today's practical. It is so your next practical is bioessay, and it has been divided into two parts. So in the first part, that is today, bioessay 1, what we'll discuss is just a brief introduction of bioessay and what is a DRC, a dose response curve, and what are its applications, and how to perform a bioessay. That is what we do in our labs. And in the second part, we'll discuss in detail what are the different types of bioessay, and we'll give you some tracings, and that will be discussed in the second part. So this is just an introductory class to bioessay and dose response curve. And the questions that generally why this practical is important, because you will definitely be asked questions about this, about the potency, about the DRCs in your exams. Okay, you may be asked in your theory also. In practical, definitely you are asked, one exercise is given, or you can have it in your spotters, or even in your experimental viva. So what is bioessay? I think you guys have done this in your physiology class. So first of all, bioessay is two words, bio and essay. So what are essays? So we define essay as essay word as a qualitative or quantitative estimation of activity of any substance. Okay. So essay basically means measuring. So whenever you are measuring qualitatively or quantitatively any activity of any substance, that is known as assay in our field. And what is a drug assays? That is, you are measuring the, estimating the activity of drug. In place of a substance, you will have a drug. So every drug will have some activity. Now what are various types or methods by which you can measure the or estimate the activity of a drug? Okay, first is a physicochemical assay. Physicochemical assay, which it's like which uses chemical methods or physical methods like spectrometry, chromatography, mass spectrometry. So these are used to measure the concentration of the drug present in a given sample, and also the structure of the drug, and can be used for, and concentration, the function is activity of the drug is considered as a function of the concentration of the drug. So if you know the concentration, then you if you know the concentration, higher the concentration, higher the activity. So these physicochemical assays, they are used, they can be used to measure the indirectly measure the activity of the substance. Next are immunological assays. There are methods which involve antigen antibody binding, like radioimmunoassays. They are used to measure the basically what you add a radio labeled isotope to an substance, and you can measure its radioactivity, or you can attach it to an antigen or antibody. So such kind of assays are known as immunological assays. The last one is your bioassays, okay, which we are going to discuss. So assay, as we discussed, they are qualitative or quantitative estimation of activity of a substance. And bio word as such means when you are doing it in a biological system. Okay, this biological system, any living system will be considered as a biological system. Okay, anything, it may be a one cell, it may be an isolated tissue, it may be a cell line, it may be microorganisms, or you can do it on a whole organism. So any method or any assay method where you are measuring the concentration of the drug or the activity, you are estimating the activity of the substance in a biological system, we call it as a bioassay. So even when you are doing it in cell culture, in microorganisms, all that, suppose you are studying the inhibition of microbial growth by an antibiotic, so that will be a type of bioassay. So there are variety, even when you are testing a drug in a human, studying the pharmacokinetics, that is also a biological assay. But what we are going to emphasize on, the assays which we do on isolated tissues. Okay, so what discussion we are going to follow is about the assays which are done on the isolated tissues. Okay, so it was first done in 18th century by the man Paul Ehrlich, and what he did, he first standardized the diphtheria antitoxin. That is, how much antitoxin is neutralizing the diphtheria toxin, the toxin produced by the diphtheria bacilli. So this was the first bioassay done, and the credit goes to Paul Ehrlich. Okay. So what we will discuss now is mainly about the isolated tissue. The bioassays which are done in our labs are the one which are done on isolated tissue, but I want you to remember that anything you do in a living system is a type of bioassay. Okay. Now what are the principles? Okay, so your physical and chemical methods, they are like, they use some physical principles, some they have a totally different basis, and your bioassay has a totally different basis. So one of the first, that is, what are the conditions under where you can do bioassay, or you can rely on a bioassay. So first is that when you are doing the activity of an unknown drug, okay, it should always be compared with an internationally accepted standard. Okay, you cannot, especially if you are measuring the concentration, then you are always comparing it with a known substance. Okay, you just cannot do it unless until there is a comparator, it has got no value. So this is the first principle. It's like when you are using a weighing balance. Okay, so on one side you must have seen the balance which is present with the vendors generally. So they have a 1 kg or 500 gram weight on one side, and they on the other side they put a equal amount of substance, so it should ultimately get balanced. Then you know that the jo bhi vegetable or fruit we have put, that is 500 grams or that is 1 kg. So this the weight that they have, that is the standard, that is and you are comparing it, the unknown weight is the, that is the unknown drug, and ultimately you are balancing the both. So bioassay is also based on the same principle, that you have a known substance and you are comparing the unknown, the activity of unknown with that known substance. Okay. Basically what we determine is the potency of the drug, and potency is a relative term which I want you to understand. You cannot say, if I ask you what is the potency of X drug, so you cannot comment on the potency of X drug until, so you have to ask compared to what, like something can be more potent or less potent. There is, as such, there is no value for the potency of any drug. Okay. And the biological activity which is exhibited by the next principle is the biological activity which is exhibited by the test compound in the living system should closely resemble the therapeutic activity of the drug. That is what for what therapeutic use we are going to use the drug, the biological activity should somewhere correlate to that. Okay, for example, if you are studying the effect of an oral hypoglycemic, so okay, you are not measuring the glucose level directly, that what is the you given a drug and how much glucose it is lowering, that we are not measuring, but it should, the effect which we are measuring should be correlated with the lowering of glucose. Like for example, in rabbits, what we do, we measure the dose at which the convulsions occur. So we know that convulsions may occur due to hypoglycemia. So this is correlated, the what activity we are measuring is correlated with the therapeutic activity of the drug. And again, the third principle, species which show maximum sensitivity should be selected for the bioassay. Okay, so for any test, when you are selecting a species, it is very important that you select the species which have maximum sensitivity. Like suppose if you are screening a drug for an anticholinergic activity, that is, we want to see how much muscle contraction on the GI smooth muscles it is producing. So that's when you are selecting a species, that substance, that species, that tissue should have some sensitivity to acetylcholine. If there are no acetylcholine receptors, no cholinergic receptors, then there is no point of doing the screening for anticholinergic activity on that tissue. Okay, so if you are selecting, suppose if you are doing for anti-serotonin activity, so we know that serotonin receptors are present on the fundus of the stomach, or the, so where the receptors are present, where the tissue which has sensitivity to that substance, only that tissue should be selected, and that's why that species should be selected. Okay, next is, and the bioassay method should give reproducible results. So this is like a principle of every method that you use to estimate the activity, that you should that the method should always give reproducible results. That is, if somebody is doing it, then the method should be such that if somebody else wants to reproduce my experiment, then they can do it and they can achieve the same result what I have obtained. Like suppose if one person did bioassay for a drug X and he found that it has anticholinergic activity, somebody else did and he found that it has some anticonvulsant activity. So it should not happen. So it should give reproducible results. That is the way in which the method should be selected. Now when do you do bioassay? Now it is very complicated procedure. We have all the physical and chemical methods nowadays. So why will you want to do a bioassay at all? So first of all, the first indication would be to estimate the unknown chemical composition with a specific biological action. Like for example, you have some extract. Okay, you don't know the exact ingredient of the extract. It can be a mixture of multiple ingredients. So, but it has a specific biological action. So you can screen the the wherever the chemical composition is not known, you cannot use a chemical assay or the physical assay to measure the concentration. There you have to rely on what activity it is producing. Okay, for example, certain plants extract, they have multiple substances. So if you go on measuring the concentration by spectrophotometry or chromatography, you will get multiple components and they have multiple concentrations, but all together as an extract, that substance is producing one effect. So there you will have to do a bioassay because you want to estimate the effect. And similarly with posterior pituitary extract, posterior pituitary extract can be mixture of oxytocin, vasopressin. So you cannot estimate the exact, you don't know the exact chemical composition. Okay, you just know the biological action, then you will go for a bioassay. Next is, if the chemical assay is very complex or highly insensitive. What do you mean by insensitive is that when you're doing the assay, okay, it may not give you the same result every time. Okay, so it mainly applies to the biological molecules like acetylcholine or insulin, although nowadays very recently chemical assays for insulin are used. Until date biological assays were used to estimate the concentration of, because you don't know the concentration of insulin, that if you have seen biological drugs, they are always defined in units, that 1 unit of this hormone, 1 unit of insulin, 1 unit of any enzyme, 1 unit of, so it is defined in the terms of activity. It is not defined, the unit is always defined in the terms of activity. It is not defined in terms of concentration. Okay, because the in case of biological drugs, it also depends on the source from where you are extracting it. So when the chemical assays are insensitive or they are highly complex, the examples would be mainly the biological drugs. Okay, again the next example would be if a drug is a complex mixture of substances of varying structure and activity. Similar to the first indication, here it is you don't know the chemical composition, and here although you know the chemical composition, but you know it is a mixture of substances of different types, especially in the plant extracts or in the posterior pituitary extract. Okay, and they have, all of them have different structure and different activity. So if you go on measuring them with chemical methods, you may not it may not reflect directly the activity of that substance. So it's always better to go with the bioassay in such substances. Okay, but there can be examples like where drugs which differ in their composition but have same pharmacological action, because at the end of the day we are concerned with the activity of the drug. Okay, what it is made up of, it may be different substances. So it is like for example cardiac glycosides. So all of them can be different type of glycosides. So it's not possible to measure, or it is not relevant to measure the concentration of such substances. What is important is what effect they are producing in the body. So in such cases also bioassay is the method of choice. Okay, because they have the same pharmacological action. Similarly with catecholamines. So you have different types of catecholamines, and there are different structures of the drugs and there are different substances, but they have the same pharmacological action, so they will produce the same effect. So in such cases you always go with bioassay. Okay, when the other indication would be when the active principle cannot be easily isolated. Okay, so because for example in thyroid glands, you cannot easily isolate the you cannot easily isolate the active principle. Okay, or you do not know the active principle. So as you can see, the major examples they involve mainly the biological biomolecules, the drugs which are biomolecules or biological drugs. For chemical drugs which have a known chemical structure, we generally go for chemical assays, but for these biological drugs, the main thing that we are concerned with is the activity of that substance. Concentration may vary, and concentration is not a reliable indicator of the activity of such substances. So that's why their concentration is always defined in terms of activity inside the biological system. And when you are defining something in terms of activity, not in the terms of molecular weight or molecular like for heparin or insulin, such in case of such drugs, the main thing is the activity it is producing. So you always define them in terms of units, not grams or not milligrams. And in such cases, bioassay is the standard method of choice to estimate the activity. So depending on the indications, they will what are the applications of bioassay, as we have already discussed. To you can use it to evaluate the pharmacological activity of new substances. Okay, like the screening of new drugs, for the screening of activity of new drugs on biological tissues. It can be used to determine the concentration of known substances in tissue extract or body fluids. Like for example, you can determine the concentration of substance in a biological sample, like for example of acetylcholine, histamine, these are or other catecholamines. So they can be you can determine the concentration comparing it with a standard substance. It can be used for estimating the biological variation of the drug to species and other group of animals or living beings. Okay. That is the same amount of drug is producing different effect in different species or different individuals. So you always have a biological variation, and you can estimate that using bioassay. Similarly, it can be used to measure ED50 or LD50 of a drug, because no chemical method can determine the ED50 or the LD50. ED50 means that is the effective dose, 50 or the lethal dose. So at the dose at which the 50% of population is dying. So for that you need a biological system to measure this. Also, standardization of drugs for obtained from natural sources which cannot be obtained in a chemically pure form, as we discussed about insulin. Okay, so for example, it is used to standardize insulin. So when I say you that 1 unit of insulin, so although now with the recent recombinant insulin preparations, insulin is defined in terms of weight. But earlier, if I asked you what is 1 unit of insulin, how much it is, like it is 1 gram, 2 gram, 3 grams, so that was not standardized. Okay, and it is the same is the case with the multiple biological molecules like heparin. If I ask you what is 1 unit of heparin, then you cannot define it in terms of weight or the molecular weight. It is always defined in terms of biological activity. So for insulin, it was something that the amount of insulin which will, I don't remember the exact values, but the amount of insulin which causes hypoglycemia, which reduces the blood sugar level by a certain amount in a rabbit, in an overnight fasted rabbit. Something, I don't know the exact amount, but the insulin which is reducing the blood sugar level by a certain amount. So it was defined in that terms, and that is how the preparations were standardized. Okay, that this is 100 unit of insulin, this is 1000 units of insulin. So in for such cases, standardization, you always need a biological assay. It is not standardized based on the weight. And the same is for biological standardization of drugs like oxytocin, insulin, heparin. So this is for drugs from natural sources. It may be from plant sources, and this is for the drugs which are biomolecules. So in general the use will be wherever you are not able to quantify the activity of the drug is not a reflection of the exact molecular weight of the drug, rather it is a reflection of the sorry, what I mean to say is that the basic application of bioassay would be in substances where activity is more is the main area of concern and not the exact concentration of the drug. Okay, it may not be that way also. In some drugs it is related also, but for most of the drugs where the activity is the main area of concern and not the exact chemical composition or the chemical structure. So it will mainly include your biological molecules. Now what are the limitations of these methods? First of all, there are ethical issues. Of course, whenever you are using a biological, an animal or a biological system, you need to get it from, even if you are using an isolated tissue, you need to get it from some animal. So there are always going to be ethical issues, that it can be unethical in some cases. Okay, and of course it is more laborious and time consuming because of all the other method that we'll discuss now, that you have to mount it on a tissue assembly, organ bath assembly, and you have to continuously study the effect. And it's also skill dependent, that you need to have a certain level of skill. It is not like the chemical methods where the machine is doing the majority of the work. Sorry. And also you'll have a lot of biological variation. Now what is the experiment that we generally do? So the aim of our experiment is, at your level, it is to make the dose response curve of acetylcholine by using guinea pig ileum preparation. Acetylcholine is one of the substances for which you do bioassay. Okay, so you have to make a dose response curve of acetylcholine, and what are the requirements? So what will you require for this? First is a organ bath assembly. Next is physiological salt solution, which is tyrol. Third is your a tissue, that is here it is the guinea pig ileum, and the tissue should be fasted overnight. Okay, and then last is the drug. Okay, so drug here we are doing for acetylcholine, so you need a acetylcholine stock solution. Okay, now we'll discuss all this one by one. So basically we are going to discuss the method of exact method. You don't have to do the experiment, but you are expected to interpret the result and of the experiment. So first of all, what is organ bath assembly? So it is a system where you are going to mount the biological tissue and you are going to study on that. Basically it is assembly, it is a bath assembly of the organ. So this is a general schematic diagram of the organ bath assembly. It is present in our laboratories. You can come and see that if you ever get a chance. And so it consists of, you can see, first of all you have to see this big big bath, organ bath. This is the organ bath chamber. Okay, here is your this test tube. Here you are going to tie the tissue. I am just giving you a rough overview of the organ bath assembly. So there is an outer bath, there is an inner bath where you are going to tie the tissue. Okay, now this, when you have a tissue, it must be aerated. So this inner organ bath is connected, is given connected with an aeration tube, that is, it is supplying oxygen, and also it is connected to a physiological salt solution. Physiological salt solution is needed for the tissue to survive outside the body. Okay, and now here is a water outlet in this organ bath, which is used to drain the water, and an outlet in the inner organ bath also, which is used to change the physiological continuous changing of the physiological salt solution. Now this is mounting of the tissue. Now here is a stirrer. So what is the role of the stirrer? And here is a heater and thermostat. So basically the heater will heat the bath water in the outer organ bath. This stirrer is going to stir the water so that the temperature is constant over the entire organ bath and not just in this area. And then here is a thermostat. The thermostat is required to maintain the temperature at a constant level, and it will automatically cut off the heater if the temperature goes above a certain level. So this is the organ bath, and now here we have tied the tissue. And what do we tie the tissue onto? So this is a liver. Okay, so this is a liver. Here is a fulcrum, and it is attached to the tissue is tied to this. So whenever this tissue is going to contract, this liver is going to swing upwards. And here we have for recording this movement, we have attached this to a kymograph drum, with a rotating drum with a kymograph. So it will record the observation. There is a stylus which will record the movement of this liver over this paper. Okay, and here is the assembly for this rotating drum. You can rotate the speed, and also you can switch it on or switch it off when you want it to stop. Now we'll see, it might be looking very mixed up to you, but if you see it in practical inside the lab, then you will be able to appreciate it better. So we'll just break it down into parts. So basically, as I told you, this is the organ bath, the outer organ bath. It is from, it starts from here. I'm sure you all have done this in your physiology. If not, you can come and see it in our department. So this bigger one is the outer bath, and it is made up of glass or perspex, and basically its main function is to store water outside the inner organ bath, and this water is used to maintain the temperature of the inner organ bath, that is this tube. This one is the inner organ bath. Okay, so this is placed inside the outer organ bath. Thus what happens, whatever is the temperature of outer organ bath, it will be reflected into this. That will become the temperature of this inner organ bath. And here the tissue is tied. So it is made up of glass to observe the tissue during the experiment, and it generally has a capacity of 5 to 50 ml. Okay, and here what you have to do is, you have to fill the physiological salt solution, and you have to do a marking at some level, so that whenever you change the physiological salt solution, you fill it back to the same level, so that there is no change, major change in the repeated observations. Okay, so generally we do marking somewhere over here, and it is used to hold the tissue. The main next is the tissue holder and oxygen supply. So this tissue holder is present inside the inner organ bath, and to which the tissue is attached. Okay, and the tissue holder is also connected to a aerator, which supplies which is responsible for air supply. Okay, so there is outer organ bath, there is inner organ bath with tissue holder and oxygen supply. So what is this motor is going to supply is air. It may give you air, the preferable. So we never give it 100% oxygen, because what 100% oxygen can first of all cause oxidative stress in the tissue, and it may make your pH of the inner organ bath, it can be made alkaline because of lack of carbon dioxide. Okay, because the tissue will keep on using the bicarbonate ions for carbon dioxide, so it will ultimately become alkaline. So that's why we use a mixture of oxygen and carbon dioxide, not pure oxygen. So this part is the kymograph, the rotating drum and the kymograph. This drum is also known as Sherrington rotating drum, and there is a gear and lever which is used to adjust the speed. Okay, so the speed of drum is 0.25 millimeter per second. And kymogram is the paper which is used to record the response of the tissue or the muscle against the known or unknown concentration of drug. And the paper is generally smoked with burning cotton soaked in benzene or kerosene. So here we also have a smoking room. If you want to see this, you'll have to come and see that the paper is generally smoked on the on the smooth side, and that is a layer of smoke is put over the paper. You must have seen this paper in physiology also, and it is pasted over the your drum, so that when the stylus moves, it can record the observations. Okay, but nowadays in many laboratories you can find a stylus with ink, so you don't need to do the smoking and all. But here in our lab we are still doing it. The next part is the liver. So liver is nothing but this it is straw or made up of aluminium or stainless steel, and at one end it has a stylus attached to it. So this is liver type 1 liver, where the fulcrum is in the between. Okay, and here is the tissue acts as the load, and this will act as so there are two arms, the effort and the load. So sorry, the tissue will act as the effort, and the stylus will act as the load. So where the effort is put, the load arm will shift upward. So here when you are adjusting the liver, you have to maintain a magnification. What is magnification? Basically it is a distance between this, the ratio of distance between this and distance between this point, that is from the fulcrum to where the tissue is dyed, and from the fulcrum to the stylus. Okay, so this is A, this is B, then magnification will be A by B. So this distance, more will be the magnification. So basically when you increase the magnification, the height of your responses goes on increasing. That is, even because your tissue is going to contract in millimeters. Okay, but you will not be able to appreciate the difference in different responses. So that's why we need a magnification. That is, even if when the tissue is contracting in a very small length, the stylus is going to move in a higher length. Okay, so that's why we get a good height of response. And why that good height is needed is because it will help you in differentiating, in grading the responses, because in practical the tissue is responding, the difference in between two responses, if you directly see the tissue, it is going to be in micrometers or something. But when you are magnifying the same response, when you can appreciate the difference or the grading of the response in the your final result. Now this was the organ bath assembly. So here you will definitely have to come to the department to see this organ bath assembly, and if possible we will definitely try to revise this, because it's not possible to appreciate it on PowerPoint unless you see it in the practical. And you will always be asked a spotter or questions over this, that what is the function of which of the parts. So you have to know this. Now next is the physiological salt solution. Now what we put inside your inner organ bar, that is the physiological salt solution. So what is its role? It is needed to keep the tissue alive, in simple words. Okay, because you have detached the tissue from its blood supply, and so we have arranged for the oxygenation, but you also need some maintenance of the homeostasis. So it is a salt solution, and so what we use for guinea pig ileum is tyrol. So there are other types of physiological salt solution also, but generally when we are using a muscle preparation, we use tyrol. When we use a nerve muscle preparation, that is a tissue which is still innervated, like in frog heart or something, there we use Krebs solution. Here we use tyrol solution. So what is the composition? I will not bother you with the exact percentage of all this, because you need not know this. What you should know is it contains all these salts made in the distilled water. So first is sodium chloride, next is sodium bicarbonate, next is sodium dihydrogen phosphate, next is potassium chloride, magnesium chloride, glucose and calcium chloride. Need not know the exact percentage of each and everything, but at least you should know the name of the salts. And what are the ions which are produced by this? So first of all, by sodium bicarbonate and sodium dihydrogen phosphate, they are simply buffer. They used to maintain the pH. What is the pH that should be maintained? It is same as the pH of your blood, that is 7.2 to 7.4. Okay, so these two are used for maintaining the pH, or they act as the buffer. What is the role of glucose? Of course, glucose is the major nutrient for the tissue. Okay. Next, what are the electrolytes which are present? First is the sodium ion. Next is potassium ion, comes from the potassium chloride. Next is your calcium ions, come from calcium chloride. Next is magnesium, which comes from magnesium chloride. Okay, and this I already told you, buffer, and it's a source of nutrient for the tissue. So what are the role of these ions? Same as their role inside the body. Why do we need sodium? Sodium is your major extracellular cation inside your body. So it is used to maintain the osmolarity of the solution and keeps it isotonic. Same as the role of sodium in your body. Next is potassium. So what is the role of potassium? Potassium is the major intracellular cation in the body. So it is important in nerve conduction. If you are using a nerve muscle preparation, it is important in muscle contraction. If you are using a muscle preparation, or even in a cardiac muscle, it is used for muscle contraction. Next is calcium ion. So what does calcium do? So we all know calcium is responsible for the excitation contraction of the muscle, nerves or glands. Okay, it is basically for, in case of muscle, it is for excitation contraction coupling. In nerves it is used for conduction. Okay, and glands it is used again for the release of the substance. Next is magnesium. So what is the role of magnesium? Magnesium is used to relax the tissue. That is, whenever you detach the tissue will always have some spontaneous activity of its own, because there must be some acetylcholine which is still present inside that portion of the tissue which you have cut. So it will always have some spontaneous activity of its own, it will keep on contracting. So we want to eliminate that for a perfect result. So that's why we add magnesium, that will reduce the spontaneous contraction, and thus you will have the baseline, you will have a straight baseline. So what is the tissue? So this is the simple anatomy. Here is the ileum. You take a overnight fasted guinea pig ileum, because fasting will reduce the spontaneous activity of the ileum tissue. And here you will cut the ileum. I will not go with the anatomical details for you, that from where you excise the ileum. So you will take a portion, you will put that ileum tissue in the salt solution. in a physiological thyroid solution, and there you will tie thread to both the ends, and then you will tie it inside the organ bath, inner organ bath, and then you will leave it there. First, when you leave it there, then when you tie the tissue will have, despite all the precautions, it will have some spontaneous activity of its own. So you need to give some time to the tissue to relax, okay, to exhaust its spontaneous activity. After some time, maybe 1 or 2 hours, your tissue will be totally relaxed and you will get a straight baseline. In the meantime, what we do, the fourth thing we need is a stock solution, that is the standard drug. So in case of this experiment we are just explaining you as an example or as a prototype. So here the stock solution we are using is of acetylcholine. So what are we going to do? What do we want? We want a dose response curve. So we want to have increasing concentrations, okay. Basically, how do we obtain dose response curve? You keep on increasing the dose and you keep on seeing the response, and whatever the graph comes, that is your dose response curve. So how will you increase the dose? So first we will prepare a standard solution which will contain 1 mg per ml. It may not be 1 mg per ml. In this experiment we are using it as 1 mg per ml concentration or 10 to the power 6 nanogram per ml, the same thing. So we will prepare this solution of acetylcholine. We have acetylcholine powder which comes as standard packet, and then we mix it in thyroid solution. Why thyroid? Because we are going to put that drug or that amount of substance inside the inner organ bath. So inside the inner organ bath you don't put anything except thyroid, okay, because the tissue needs that to survive. Now what we do, we make serial dilutions by a factor of 10, okay. So what happens when you are diluting the 1 mg per ml solution? When you are diluting, the strength will go on decreasing, the concentration. So suppose this was 1 mg per ml or 10 to the power 6 nanogram per ml. I took 1 ml of this and diluted it with 9 ml of thyroid. So what happened? It got diluted 10 times. So the concentration will become 1/10 of the original concentration, that is 10 to the power 5. Now I took 1 ml of this, again I mixed it with 9 ml of thyroid. Then what happened? Again it got diluted 10 times. That is, getting diluted means the concentration is decreasing. So the concentration decreased from the previous one by 1/10. That is, if this was 10 to the power 5, it became 10 to the power 4. So we keep on repeating this, okay, and we make a solution of 1 nanogram per ml. Now how do we decide that what dilution we have to, how many times we have to dilute it? It is decided depending on the sensitivity of the tissue. That is, we should know that at what is the sensitivity of the guinea pig ileum for acetylcholine. Depending on that, we have to make the solution of that. The most diluted solution will be of that concentration, okay, because we have to start from the lowest level. So this is just to make you understand, you don't have to do this. And thus these are our dose preparations, okay. We label them and keep them aside. Now there is one more terminology, dose cycle and response. So what is dose cycle? So what happens, you give one dose, okay, then the tissue will go on contracting, again it will come down to the baseline, and you need to let it move for some time, okay, at the baseline. So this is the, I'll show you in the graph. So the time and then you add the next dose. So this is the dose cycle, is the time gap between the drug dose additions, And what is contact time? Contact time is the time for which the tissue comes in contact with the drug. Okay, it will be a part of this dose cycle. The dose cycle will have three phases. First there will be a baseline recording, then there will be dose response, and then you stop your chymogram, the drum there, and then you wash your tissue. So see, this is the baseline. First you let the drum move, record a baseline proper. Here what we injected the drug, the first dose, you got a response, and then you washed the tissue. So you allowed the you allowed the stylus to come down after the response, and then here you stop it, and then you wash the tissue before giving the next dose. Okay. So what is the duration of the dose cycle? It depends on the type of tissue. So there are two types of tissues: slow contracting tissues and fast contracting tissue. So what is slow contracting tissue? It has nothing to do with your slow muscle fibers, fast muscle fibers. Okay, these are types of tissue which have been found to have different rate of contraction. The examples of slow contracting tissue is frog rectus abdominis, guinea pig tracheal smooth muscle. The fast contracting tissues are ileum, uterus, ascending and descending colon. Okay, so in fast contracting tissue they will contract fast. The slow contracting tissue will contract slow. So here you need to have a longer dose cycle to record a proper response. So here the dose cycle is of 5 minutes, and the contact time, that is this time, should be of at least 1 to 1.5 minutes. And fast they will keep on contracting very fast. So here the dose cycle should be of 3 minutes, and the contact time should be not more than 30 seconds. You should record the response should come, because they are contracting fast. Basic logic. So this is an actual picture of a result that we have obtained in our experiment. So you can see this is the first dose, second, third, fourth, fifth, sixth, seventh, eighth, ninth. So this is the threshold dose, at the dose at which the first response comes, and there will be a maximal dose. That is the dose after which, no matter how much you increase the concentration, the response will not increase. So this mainly occurs because of the saturation of or the down regulation of the receptors. So how do you go on increasing the dose? Just showing you this table, because you can be asked this in the exam. You are given table and you are asked to complete it. So when I gave you so the first dose we started was with 0.1 ml of S into 10 to the power minus 4. That is, it contains 10 nanogram. Just go previous to this one. See, here you will have, I know you haven't paid attention, but so it is 10 to the power minus 4 contains 100 nanogram of, if you are diluting this 1 mg per ml. Okay, so I am giving 0.1 ml of 100 nanogram. So I am giving 10 nanogram of dose. Similarly, what I did, I kept on doubling the dose. Okay, so when I came at 0.8 of S2 into 10 to the power minus 4, when it should become 1.6 of this, the double. So instead of 1.6 of this, I gave 0.16 of 10 to the power minus 3. So just we are doubling the dose, okay, consequently, and doubling the volume, and the dose is also automatically getting doubled. From 10 it became 20, 20, 40, 40, 80, 80. Okay, this. Now what we do, we calculate log of the dose. Why? Because the dose is doubling. Okay, so we have to plot a graph, and you know that for dose response curve we try to plot a graph, we'll not get a true graph, because your dose is increasing in the geometric progression. Okay, it is increasing in the logarithmic fashion, it is getting multiplied by 2. So in such cases, wherever your variable is increasing geometrically, in geometric progression, not arithmetic progression, then you have to use a logarithm of that. Okay, so that's why what we do is we plot a log DRC, not exact DRC. So we take the log dose of all the individuals dose. So this is the value of the log dose, and then we measure what response we got from the tissue at the particular concentration in millimeters. So we measure it with the scale on that paper, that this was the response. So from this point to this point we measure the length of the response. So this is the length of the response. Okay, so you can see we stopped the experiment when we got two similar responses at double dose. Okay. Okay, and now using this response, log dose versus response, we plot the DRC. So brief introduction to dose response curve. What is DRC? It gives the relationship between the dose and the effect of the drug, and the dose response data are typically graphed with the dose on the x-axis and the response is on the y-axis. Now we can plot two types of responses: dose versus response or log dose versus response. So what will happen if I plot dose versus the response? How will you plot this on the graph? The dose goes from 10 to 20, 20, 40, 40 to 80. Okay, so it's not possible on the normal graph paper, because as I told you, it is getting doubled, increasing in logarithmic fashion or geometric progression. So that's why we use log dose of such calculation. So generally this is a DRC which we have plotted. So as we see a graded effect, that is, as the dose is increased, the response will also be increased until, so it is an S-type curve. Hope you all have read dose response curve in the theory classes, and the response will keep on increasing till the maximum response is reached. And the slope will, what does the slope denotes? It denotes the change in response per unit dose. Okay, so you can see we plotted on the log dose versus response. Now what is ED50? ED50 is the dose that produces 50% of the maximal response. Okay, so this is the the maximal response was at 8.2. So the half of the 50% of that is at 4.1. Okay, so here what concentration produces this response? You can plot it on the graph. It is 2.2. So you have to, this is log of the actual dose. So you have to calculate the anti-log of this. It will be 158.5 nanogram. Okay, so that will be the EC50 of this particular substance. Okay, see, so that reflects the, if less is the EC50, more is the potency. But I have seen in some books it is written that this is defined, EC50 is defined as the potency. So I want to tell you, potency is always a relative term. You cannot define potency for any substance. You can only say it is more potent or it is less potent. You cannot say that its potency is 158.5 nanogram. No, that is wrong. Okay, potency is always, it is more potent than a drug whose EC50 would be 170. Okay, it is more potent than that, but you cannot say that this is potency, this is EC50. Now I told you this is the threshold dose. It is the minimum dose which produces measurable response. So what we generally do is we when we get a response, we repeat the same dose just to say confirm that this response was not by chance. So we got this, this is known as standardization of the response. So we repeat it twice to ensure that we get the same response. Then we started doubling the dose. So and the maximal dose is here, the response which produces, the dose which produces maximal attainable response. You can get graphs and you can be asked that identify the threshold dose or the maximal dose. So here is an exercise for you. Okay, there was a bioassay which was conducted on isolated guinea pig ileum tissue. See, if you have any confusion about what we discussed till now, I am sure it's not very clear. We do bioassay for as a part of our postgraduate teaching. So you can always come and see the preparation or the response. If you want to see graphs, we'll discuss it in the next part. Right now, if you can solve this, I think my part is done. So it was conducted, there was a bioassay experiment which was conducted in isolated guinea pig ileum tissue. Okay, here the stock solution is 10 mg per ml. In our experiment, we did it as 1 mg per ml. Okay, so and these are the values which are already given, and this is the hypothetical observation table. So you have to complete this table. First of all, what will be the first dose? We start with 0.1 ml of S2. So this is already given, you have to complete this. So can you do that? So first of all, what will be the unit of this? See, what will be the first dose? If you can calculate this, you can keep on doubling it. So here standard solution is 1 mg. Oh, I'm sorry, here it is given 10 mg, it should be 1 mg per ml. Okay, so the standard solution is 1 mg per ml. So it is, the strength is S by 1000. So what it will become? 1 microgram per ml, right? Now we are giving 1 microgram is equal to 1000 nanogram. Now but we are giving 0.1. So 1 ml contains 1000 nanogram. We are giving 0.1 ml, that will contain 100 nanogram. So that is going to be our first concentration or the dose, 100 nanogram. Now what will be 0.2? You'll have to double it, 200. What will be 0.4? 400. Again we have done 0.4. Why? Because this was our threshold dose, the first response. So we standardized it by repeating. So this will again be 400. This will be 800 nanogram. What will be 0.2 ml of this S by 100? See, this was 200, so it will become 20 nanogram, 40 nanogram, 80 nanogram. Okay, this will become 2 nanogram, 4 nanogram, 8 nanogram. Okay, so you have to complete this table. You will be asked this question. Complete the table. Identify the threshold and maximum dose. What is the threshold dose? The dose at which the first response occurs. That is this one. So whatever value comes in your this box, that will be threshold dose. Okay, and what will be a maximal dose? Whatever value comes in this one. Okay, because here now the response is constant. 28 is the maximum response. Here also, because so this is your 0.2 ml is your of S by 10 is maximal response. So whatever value comes in this box, that will be a maximal dose. Now next will be plot the log dose response curve using the observation table. So what you will have to do is, you got the values in this table. Once you have filled them, you can calculate log of these values. So what will be log of 100? It will be 2, and similarly log of 200 will be log 2 plus 2. So we have already given you the value of log 2, log 4, log 8. You will have to calculate the value of all these log values of all these values which were obtained, and that you will plot on the x-axis, and the response is given here in millimeter, that you will plot on the y-axis, that will give you your DRC. Okay, and here we are asking threshold and maximal dose in micrograms. So micrograms from nanograms you have to convert it into micrograms. So you will be asked this question, not exactly same values but similar question in a practical exam. So this is it for now. Then next, the types of bioassay, some details or anything if you have not understood we will be discussing in part 2. So if you have any question right now, you can just type the message. Anyone has any question? Okay, so rest everything we'll discuss in the next part, that is part 2. Thank you.